let's talk about the most personal instruction manual ever written. I mean, the actual source code that makes you, well, you. We're diving into the human genome, the literal blueprint for life.
And hey, this isn't some far-off science fiction. This is the world we live in right now. Understanding our genetic code has thrown open doors we never thought possible, and it's changing everything from your doctor's visit to the crime lab.
So, to really wrap our heads around how all this is even possible, we got to start at square one. What exactly is a genome?
Think of it as the ultimate master blueprint. Seriously, every single instruction needed to build and operate a human being, from your eye color to how your cells fight off a cold, it's all written in this code. It is the source code for life.
But, not all blueprints are drawn the same. The complexity of a living thing is totally reflected in how its genome is put together. So, let's take a look at the simple versus the complex. Right away, you can see a huge difference. You've got your simple organisms, prokaryotes like bacteria. Their genome is this neat, compact, circular thing. It's all business, just packed with genes, super efficient. But then you look at a complex eukaryote. That's us. And our blueprint is totally different. We have these massive linear chromosomes all tucked away inside a nucleus. And the craziest part? It's filled with these huge stretches of what scientists used to call junk DNA. Turns out, it's not junk at all. A lot of it is absolutely crucial for telling our genes when to turn on and when to shut off.
Okay, so that brings up a pretty big question. How in the world do you solve that packaging problem? How do you cram literal miles of DNA into a space smaller than a speck of dust? Well, nature came up with this brilliant system. Our DNA wraps itself around these proteins called histones, kind of like winding thread around millions of tiny spools. This whole complex is called chromatin, and it's just this incredibly elegant solution that keeps everything from becoming a catastrophic tangled mess.
So having the blueprint is one thing, but being able to actually read it, that's a whole other ballgame. And that's where DNA sequencing comes in. It's our Rosetta Stone, the tech that lets us finally translate the language of life. Our first real breakthrough for reading DNA came back in the 70s with a method called Sanger sequencing. Now, it was painstaking, it was slow, but oh man, was it revolutionary. For the very first time, we could read the precise order of the genetic letters.
The real genius behind this method was using these special terminator bases. So here's the trick. You make a ton of copies of a DNA strand, but these special bases act like random stop signs, halting the copying process at every single possible letter, A, C, T, or G. And each of these little terminator letters gets its own color. So when you sort all those new fragments by size, from the shortest to the longest, a laser can just read the sequence of colors. It's this incredibly clever puzzle that really laid the foundation for everything that came after.
But let's be real, Sanger sequencing was like reading a massive encyclopedia one letter at a time. The absolute game changer was next-generation sequencing, or NGS. This was like inventing a machine that could read millions of pages all at once.
The key innovation is right there on the screen, massively parallel. Instead of Sanger's slow, one-at-a-time process, NGS runs millions of sequencing reactions at the same time on a single tiny chip. This colossal leap in scale, plus a jaw-dropping drop in cost, is what took sequencing a human genome from a wild dream to a practical reality.
And the tech just keeps getting better. The latest mind-blowing stuff is nanopore sequencing. Just imagine this. You feed a single, super long strand of DNA through a microscopic hole, a nanopore, and you read the bases one by one as they squeeze through, just by measuring tiny changes in an electrical current. It's happening in real time, and it's changing the game all over again.
All of this incredible technology really culminated in one of the greatest scientific achievements in human history, the Human Genome Project. This was our biological moonshot, an audacious, unbelievable plan to read our entire genetic instruction manual from cover to cover.
So, it kicked off in 1990. It was this massive, global effort, a 15-year mission that brought scientists together from all over the world. But here's the amazing part. They didn't just pull it off. They finished early. By 2003, they delivered the first high-quality sequence of the human genome, ahead of schedule and under budget. Just incredible.
So, after all that work, what did they find? What was the final count? 3.2 billion. That's 3.2 times billion letters of code that write the instructions for a single human being. The scale of that is it's staggering to even think about.
The project's legacy is so much more than just that number. It gave us a standard reference genome to compare all other human DNA against. It totally supercharged the race to make sequencing cheaper and faster. It pretty much created the entire field of bioinformatics just to handle all the data. And it showed the world that massive, open, international science could solve humanity's biggest puzzles.
And what's truly remarkable to me is the foresight of the people who led this project. From day one, they set aside a chunk of the budget just to study the ethical, legal, and social implications, they called it ELSI, of their own work. They knew this knowledge was powerful, and they knew we had to start thinking about the consequences immediately.
And that brings us right back to the beginning, to the big picture. We've cracked the code. We've built the tools to read it. So how is this genome revolution actually changing our lives, right here, right now?
Well, first up, medicine. We can now do what's called personalized or precision medicine. By reading your genetic code, doctors can predict how you'll react to certain drugs, avoiding nasty side effects. They can pinpoint the exact gene causing a rare disease, and they can even design cancer drugs that specifically target the unique mutations in your tumor. It's a whole new world.
And then you have forensics. I mean, DNA fingerprinting has been around for decades, sure. But now, things have gotten wild. Investigators are solving decades-old cold cases by using public genealogy websites. They find a distant cousin of the suspect who uploaded their DNA for fun, and suddenly, they have a lead. It's incredible.
And this revolution isn't just about us humans. It's also about what's on our dinner plate. By sequencing the genomes of crops and animals, scientists can identify the exact genes for things like drought resistance in corn or better milk production in cows. This gives breeders a massive shortcut, letting them develop hardier crops and healthier animals way faster than ever before.
So yeah, this power to read and truly understand our own code, it's transformative. But just like the founders of the Human Genome Project knew, it also forces us as a society to ask some really tough questions.
Who owns your genetic data? Who has the right to see it? How do we make sure it can't be used against you, say by an employer or an insurance company? And how do we make absolutely sure these amazing new technologies benefit everybody and don't just widen the gap between the haves and have-nots?
And that leaves us with one last massive thought. For all of human history, we've only been able to read the book of life. But now, with technologies like CRISPR, we're learning how to pick up a pen and rewrite it. The fundamental question isn't, can we anymore? The question is, should we? And that's a conversation for all of us to have.